We explain some of the many benefits of using direct reverse transcription. Meet Priya. She's a researcher at a university who struggles to synthesize cDNA from a limited number of mammalian cells without losing yield during the RNA purification process. A colleague tells Priya about a faster, more reliable method called direct reverse transcription using proprietary Invitrogen SuperScript IV technology, which allows researchers to synthesize first-strand cDNA from samples as small as a single cell up to 10,000 cells without the need for the RNA isolation step, but generating equivalent or better results. With the Invitrogen SuperScript IV CellsDirect cDNA synthesis kit, Priya learns it can help her simplify her workflow, save time and freezer space, minimize sample loss and reduce errors, get superior sensitivity while maintaining convenience for downstream applications, including vital research for viruses like SARS-CoV-2. Let's see how it works. After preparing the cells and the lysis solution, Priya performs cell lysis and adds the stop solution. With the SuperScript IV CellsDirect kit, Priya doesn't need to isolate RNA before preparing the RT reactions. She skips multiple steps typically involved in traditional cDNA synthesis methods. Thanks to a powerful enzyme and simplified process, direct RT is done in one test tube and finished in less than half the time of traditional RT methods. Priya also doesn't need an RNA extraction kit and can complete her RT reaction in just 10 minutes, which saves her lab time and effort. Cutting out RNA isolation also helps her minimize potential sample loss and reduces room for errors, all while avoiding RNA storage in her freezer. Priya is happy with her results. The entire sample of lysed cells can be used for an RT reaction due to the SuperScript IV CellsDirect kit's superior high tolerance to common inhibitors in the cells and quick RT reaction time. This enables high cDNA yields to use in downstream applications, such as PCR and qPCR. Now that Priya understands the benefits of direct RT, she can work faster and more effectively, now that she has a method for enabling consistent, reliable results.